Mitochondria. Responsible for supplying energy in the form of ATP, it is an important organelle in our human body. However, it has its own DNA that codes for proteins, a characteristic not found in other organelles, least chloroplasts. This led us to think that mitochondria could have been cells on their own in the distant past. So how did it get trapped in our cells? A typical explanation for the occurrence is due to an endosymbiotic event, which an anaerobic nucleus-bearing eukaryote host cell engulfs the mitochondria via phagocytosis. You may wonder why the mitochondrion is not live and exists till today. This is the beauty of endosymbiosis. Think of mitochondrion as a small factory and the host as MNCs. Mitochondrion is specialized in recycling harmful waste into useful products, but it is small and lacks resources to sustain. The host is rich in resources but lacks that particular skill of mitochondrion. The result, mitochondrion will sign a contract with the host and work for them. But in exchange, it can profit from the resources supplied by the host. Putting into context, mitochondrion absorbs resources from the host and reciprocates by detoxifying oxygen into ATPs, allowing the host to thrive in oxygen-rich region. However, this traditional theory has its flaws. It assumes that mitochondrion contain only oxygen-dependent reactions and doesn't account for variations of mitochondria among all eukaryotic lineages. As of present, 4 different kinds of mitochondria exist. There are aerobic mitochondria that we just mentioned, anaerobic mitochondrion that does not process oxygen, hydrogenotomes, mitochondria that lacks electron transport chain, a complex structure that accounts for the high ATP production in normal mitochondria, and lastly mitosomes, mitochondria found in human intestinal parasites that doesn't even produce ATP. Looking back, additional theories is needed to explain how an engulfed aerobic ATP producing mitochondrion could have diverged into so many different functions. An alternative theory suggests that the host was a prokaryote instead. On the other hand, the mitochondrion is a metabolically versatile facultative anaerobe, which means that it can live with or without oxygen. The various specialized forms of mitochondria have evolved from one facultative anaerobic ancestor. From this perspective, it explains that eukaryotes evolved from the mitochondrial endosymbiosis in the prokaryotic host, accounting for the divergence of eukaryotes with mitochondria functioning differently. In summary, we have learned the origins of mitochondria and integration into eukaryotes via endosymbiotic relationship. While the traditional theory is more sounding, it doesn't explain for the varieties of mitochondria. The newer theory fits in nicely, and I believe that such understanding may give new insights on when mitochondria